sequencing, genome editing, and drug designing are the topmost techniques of bioinformatics. In this video, we will learn about drug designing, commonly called rational drug designing. Drug designing is one of the most advanced technique that is developed in bioinformatics. Drug discovery and drug development are the two other terms associated with drug designing. Basically, drug designing is developing a new drug entity, new chemical entity that never existed before, or it may be modification of the existing drug molecule, or it may be a modified method. So it may be a new chemical entity, a new drug, or new chemical altogether, or it may be a modified chemical, modified based on the pre-existing chemical, or it may be a modified method. All these are produced in the process called drug designing. It involves the scientists and a pharmaceutical company, and the drug designing process normally takes about 5 to 10 years. So it is a very expensive method. Nevertheless, in the world, several drugs have been designed and produced and released into the market. Once the drug is produced in the final stage, the drug has to get approved by FDA. One of the most common methods of drug designing is computer-aided drug designing. There are two other terms that are always associated with drug designing, that is bounded rationality, the other one is serendipity. So all these things we will learn in this video. Let us understand drug designing in little more details. Let us first define what is a drug. A drug is any chemical substance that causes a change in an organism's physiology or psychology when consumed. Drugs are different from food and nutritional products. Let us understand drug designing in 5 simple steps. It starts with the drug target, where the drug has to bind and act. This may be a gene, it may be a gene product, it may be in the organism, it may be anywhere. The drug will go and bind to this target, and then the effect is brought about. So we have to work on the basis of the drug target, on the structure of the target. So that is the first step. So the next step is there are a number of drug-like molecules which are there in various databases. They may be in plants, animals, and microbes. So we have to look for drug-like molecules. And these drug-like molecules are modified or designed in order to fit into the target. So that is the third step. In the 4 steps, the best fit molecule, the molecule that fits exactly into the target, is selected. In the final stage, in the 5th stage, the drug molecule that is selected is further developed and manufactured. What are natural drugs? The most dominant natural source of drugs is plants. Due to their chemical and structural diversity and the biodiversity of their components, so plants are the major sources of natural drugs. We have examples in the form of digitoxin, we have opium, aspirin. So these are all plant-derived drugs. What is drug discovery? It is a process through which potential new therapeutic entities are identified and discovered using a combination of conventional, computational, experimental, translational, and clinical models. The drug development is the process of bringing a new pharmaceutical drug to the market once the lead compound has been identified through the process of drug discovery and drug designing. You start with thousands of compounds and you screen and you do all your preclinical research, clinical research. Finally, you get one approved drug. It will take about 5 to 10 years for this whole process to complete, and it will cost about 800 to 2000 million US dollars. Rational drug designing is an inventive process of creating a new chemical that never existed before or finding new modifications based on the knowledge of the biological target. Rational drug designing begins with a knowledge of specific chemical responses in the body or target organism and tailoring combinations of these to fit a treatment profile. Due to the complexity of the drug designing process, two terms of interest are always associated with drug designing. They are bounded rationality and serendipity. Let us understand rational drug designing in little more detail. There are three pathways A, B, C. So each pathway begins with a different source of drug-like molecules. This is A. So you start with the compound database, and you from this you screen and get the lead molecule. Then you find out whether it is binding to the target or not by looking into the protein ligand complex, and protein ligand docking methods are used here. So once the complex is forming, then you redesign, and if it is good enough, you can produce a new chemical entity. If it is not good enough, then you go and see target enzyme receptor site and then test whether it is fitting there or not, and then you go for 3D structure, crystallography, NMR, electron microscopy studies, then do homology modeling, then you redesign the same molecule and produce the new chemical entity. So this is pathway A, which is little longer. Let us start with the B1 here. This is second pathway. Let us start with B1 here. You're starting with a 3D ligand database. We have a database where drug-like molecules are there. You directly do docking, and if it is forming a complex, then you redesign, improve, and produce the new chemical entity. The third pathway is starting with quantitative structure activity relationship 3D structures, and here we have drug-like molecules which are almost ready and good enough to form a very good complex, and once they are binding to the target, then you go for testing, redesign, and produce the new chemical entity. So these are the three normal pathways of designing a drug. Drug-like molecules are obtained by QSAR, quantitative structure activity relationship. It represents an attempt to correlate structural or property descriptions of the compounds with the activities. These physicochemical descriptions, which include parameters to account for hydrophobicity, topology, electronic properties, steric effects, are determined empirically or more recently by computational methods. Find out whether the drug is fitting into the target or not, you do molecular docking. It's a computational process widely used for rapidly predicting the binding modes and affinities of small molecules against their target molecules. This is in silico process has achieved a position of great importance in the drug discovery and drug designing process. This is the outline or schematic diagram of how protein docking is done. We will study protein docking in a separate video in detail. Once the drug is ready, the new chemical entity that is produced by drug designing process will go through various stages of preclinical trials, and once the drug is passing through all these human clinical trials, the drug will be later finally selected for further development. Let us now understand why should we do drug designing when it is such a long process and such an expensive process. Drug designing is done to produce new, novel, better drugs, to produce new chemical entities, to produce drugs for diseases for which there is no treatment or method of diagnosis or method of treating at all. Analyzing molecular modeling of genes, gene expression, gene sequence analysis, and 3D structure proteins, using in the diagnosis of diseases such as lung cancer, brain cancer, breast cancer, Alzheimer's syndrome, many such diseases for which there is no effective treatment so far. The most popular methods of drug designing nowadays is computer aided drug designing. It includes finding, developing, and analyzing medicines and related biological active compounds by computer methodologies. The use of CADD methodologies speeds up the early stages of chemical development while guiding and speeding up drug discovery process. New chemical entities are the compounds which are produced by drug designing process. They will have promising activity against a particular biological target, and hence they are useful in treating a particular disease. Certain diseases which are called orphan diseases involving fewer than 2 lakh people, and when the incidence of this disease is very low, these diseases are called orphan diseases because the drugs are not normally developed for these diseases by pharmaceutical companies, and the drugs that are developed for orphan diseases are called orphan drugs. Database of drug-like molecules from which the drug-like compounds or lead compounds are selected are proteins, and there are different forms of proteins which are available in living systems. From this you select the suitable drug-like molecule and use it for further drug designing process. The list of designer drugs, the drugs that are designed and produced and released into the market. There are many drugs that have already been designed. This is only a small sample that I have given you here in this slide. These are some of the top drug companies or pharmaceutical companies of the world, and these are top pharmaceutical companies of India which are involved in the development of drugs for various diseases in the world. And ultimately, once a drug is ready, it should get the approval from FDA, and if it is in India, it has to pass through ICMR. Drug designing is an elaborate, expensive and exhaustive process. In this video, I only have given you a little bit of introduction to the entire process of drug designing. Thank you very much for watching my videos. Please like, share and subscribe. We'll meet again in the next video with some other topic. Thank you very much.